many thanks to James and Leona for making this happen. I'm very sorry I couldn't be there in person, but glad that I could virtually join you here from Philadelphia. So you can hear me okay?
Yes.
Okay. So Bettina asked me to talk about personalized medicine, and that's what the title is. What is it, or what isn't it? And it's a big topic, and I'm going to just try to cover a few points, and this is what I'm going to go through. A couple of definitions, just so we're all in the same playing field. Really what the promise of this is, and what many of you are looking forward to in this whole area. I'm going to talk about the opportunities, but also the obstacles, and some reality check. I'm going to spend a fair amount of time talking about the scientific rigor. I'm a scientist as well as a clinician, and I think it's really important to make sure that this is all in perspective and what is realistic in the current time, the short-term future, and the long-term future, and then end with a little bit of a discussion of what the vision is. And we'll talk about a clinical trial that will be opening in Philadelphia, probably in January or February, that we're very excited about. So the first thing is that this concept of personalized medicine is not new. It really is just an individualization of care, and many of those of you who have been on the neuroblastoma road know that at the initial diagnosis, everything is very protocolized. We all sort of agree on how to approach the disease at diagnosis, but at relapse especially, you probably have seen, if you're on that road, that there has been an individualization of care. And really what we're talking about here is that the power of technology and our ability to learn a lot about the tumor through genetic technologies has made this personalization go to the next level. And so personalized genetic medicine is the practice of medicine that takes the individual's genetic profile and uses this to guide decisions that can range from prevention of disease to the diagnosis or prognostication to the treatment of the disease. And there's terminology is a little, some people use the word personalized, some use precision. It's really the same thing. And what makes things interesting in the cancer field is that personalized medicine is, for hypertension and heart disease and other conditions. But in the cancer field, there's your own genetics as well as the tumor genetics. So there's really two different personalized genomes. So on this next slide, again, some more definitions. Genetics is the study of the genes you are born with. So anyone can have their genetics studied. But this term cancer genomics is the study of how these genes become disorganized in cancer cells. And most of current genetics is focused on the DNA. DNA is what this picture is. It's 3 billion building blocks that make us what we are and how we're different. And DNA turns into what's called RNA, and that's turned into what's the proteins in our body. And when we're targeting something with a drug, it's the protein that we're targeting. So DNA is the code that lets us know what's wrong or what's right with the protein. And what's really enabled all of this and why I'm talking to you today is that there's been dramatic advancements in our ability to read out DNA sequences. When I first started in the laboratory, we were doing DNA sequencing, but it would take about a week to read out just a few thousand bases. And now we can read out all 3 billion of these in a matter of hours. And that is both exciting but also challenging. And as I already alluded to, the other thing to keep straight in all of this is that there's something called germline, which are all the genes in your body, versus the tumor, and people refer to that as somatic. And so, as I already said, cancer is a disease of two different genes or genomes. Everything that you're born with, and that's the field of understanding why do children develop cancer in the first place, and how do the genes in our body modify, why do some kids who get chemotherapy not get very sick and other kids get very, very sick. And a lot of that is encoded in the genes in our body. But much of what we're doing in terms of trying to find treatments is focused on sequencing the tumor cells themselves and seeing what has gone wrong in that process of how the tumor arises in the body and progresses to the time when it's diagnosed. So I put this in just last night. This is a paper that was published this week in the New England Journal of Medicine from my friends and colleagues at the St. Jude Children's Research Hospital, and I was privileged enough to write an editorial about it. And I think it's very important to discuss here, and maybe we'll talk about it more in the question and answer or the plenary session. But this study looked at the germline, or the genes children are born with, inherited from their parents. And the major finding of this study is that about 1 in 10 children have a genetic cause that could be detected with DNA sequencing. And this is a higher percentage than we had previously realized. And my editorial tried to put that in perspective because it's important knowledge, but we are still in an era where we have more questions than answers than how to deal with that. For children with neuroblastoma, we know that only 1 or 2% have a heritable mutation. And the take-home point for all of you in the audience is that this is a discussion you should have with your physicians. Is there a reason to look for a genetic cause that may be important for your child, your family, or your child's future? And we can talk more about that, and I'll come back to it in a little bit. So the vision of personalized neuroblastoma care is I tried to put here. I mean, and all of these things are possible. It could allow for us to provide monitoring for those who are predisposed to the disease. It could provide for much more precise prognostication or therapy assignment. It could provide for much more rational and precise therapy, a true personalization of therapy. It could provide much more refined ability to monitor the disease, to make sure it's in remission, and to monitor for relapse. And as I mentioned, it could also help us understand how to dose drugs in a much more individualized fashion. And this is something that we've achieved in some diseases, but is not yet really done in neuroblastoma. As I mentioned in the beginning, personalized medicine is a hot topic now, but it's not really new in this disease. We've known for over a decade that there are certain genes, ALK and FOX2 are their names, that predispose a child to develop neuroblastoma. Manfred Schwab in 1983 discovered McN, so that's been around for a while, and we've known for a long time that that is an important marker of disease aggressiveness. And as I'll come back to near the end, anti-GD2 therapy, which has been pursued since the 80s and has really been credentialized recently, and MIBG therapy are targeted therapy, right? They're going after specific unique neuroblastoma cells. And then in other diseases, like leukemia, there is a more nuanced approach to monitoring the disease and dosing drugs than we've achieved yet in neuroblastoma. But these things are possible, and many of us are working towards making this real in neuroblastoma in the short term. So we'll talk a little bit, a couple of times in this talk, and maybe more in the discussion, about the ALK, which stands for anaplastic lymphoma kinase. And this thick diagram is a picture of the ALK protein in the surface of the cell, and this is outside of the cell. I don't know if you can see my arrow. And this is the business part where the where an abnormal ALK will tell the cell to keep on growing, and there's a lot of work on defining ways to make drugs to inhibit this accelerator switch. So I'll come back to this, but ALK, I think, is an important example of the promise as well as the obstacles to true personalized medicine, and we'll talk about this a few times through this talk. While there's a lot of promise, there are major obstacles that everyone has to be aware of. And this is this sort of balance of the hope of this, but let's talk about what the realities are. First of all, we talked already a little bit about genetic predisposition. And when I began to study genetic predisposition over 20 years ago, I had no clue how complicated it would be. And so it is sometimes simple when you have an ALK mutation, but most children develop neuroblastoma without that sort of predisposition, and we've learned a lot about it, but we don't have the ability to do anything about it clinically yet. Moreover, when cancer starts, the genetics of the tumor cell are incredibly complex, and I'll show some examples of that in a minute. And I think of it as an ecosystem that is constantly evolving. A lot of people talk to you about, let's sequence the tumor that came out of the child at diagnosis. As I'll show you a little bit later, the tumor after treatment or at the time of relapse is completely different, and this is something we have to keep in mind as we're trying to achieve personalized therapy. And the other major challenge to the field in trying to learn about this is that the child suffers a relapse with neuroblastoma. Our tools are perfectly fine for diagnosing relapse. MIBG scans, PET scans, they're very precise. And there's never been a need to biopsy the tumor to make a diagnosis, and therefore we haven't had the critical research samples to make progress. I'll talk to you about how that has changed, and I think some opportunities we have moving forward. So this is back to the germline and how complex it is, and this is a figure from the paper I mentioned from the St. Jude group. And in yellow is neuroblastoma. So the good news is that most of these cancer predisposition genes were not mutated in children with neuroblastoma. Here's ALK, what we knew about. But the challenge is that there are mutations in other genes that are known to be cancer predisposition genes in adults. The BRCA2 is a breast cancer gene. And there were a few children with neuroblastoma, as well as a few children with rhabdomyosarcoma, brain tumors, who had mutations in these genes. And what you need to talk to your doctors about is this something that should be tested in your child? And if you discover this, what does it mean? We don't know yet whether or not this had anything to do with them developing neuroblastoma, or if it just means that they're at risk for another cancer later in life. So I bring up this very complicated field because I think it will get a lot of press, and I think you need to keep this in the context. There is still a lot that we're learning about why children develop cancer, but this is a rapidly evolving field. So in terms of the tumor genome, this is a very complicated figure, but I'm going to try to make 2 or 3 points. This is from a paper we published this summer, and what this wheel is trying to show is all of the DNA changes around the outside of the spokes of this wheel. And then listed here in these letters that you may or may not be able to read are the genes that were mutated. And this is a tumor from a child diagnosis and the same child at relapse. And these lines connecting the wheels and these colors are the chromosomal rearrangements or changes. the point here is that there's a lot more going on in the relapse on my right than diagnosis. And these bar graphs at the bottom, and can you see my arrow?
Yes. Yes.
Okay, good. So each set of bars is the same thing from diagnosis and from relapse. And if you look at this one patient, NB1269, what it's saying is that there were 40 mutations that were the same at diagnosis and relapse, but all these yellow mutations are new. And so the point of this is that, again, to reiterate, these cancers are always changing. And if we're going to make decisions about drugs matched to mutations, we really need to try to get material as close to the point of decision as is possible. And I hope that makes sense. The other major challenge we have is that neuroblastoma is quite sinister, and we're often faced as clinicians with this sort of issue. I just said to you we would like to get tumor at the time of relapse, but sometimes relapse is like this, where this little dot here is the relapse. And I think even just 2 or 3 years ago, most physicians would say it would be impossible to safely biopsy this and get DNA sequencing results. But this is an example where we have the technology now in interventional radiology to go into this exact spot to get what can be sometimes, this is the tumor here and this is all debris, and it used to be you wouldn't be able to technically get DNA sequencing results from such a small amount of material, but the technology has changed so dramatically that we can, and this is the sort of result we get, and this is a type of change in the DNA that we actually think we know how to do something about. And so this mutation leads us to a potential clinical trial. So the point of this is that things are changing pretty rapidly in the ability to safely access tumor tissue and do something about it. So the take-home message from all of this is that, in my opinion, we are now in so-called equipoise, that a child who suffers a relapse, we can potentially benefit that child, and we should consider a biopsy at the time of relapse. And I think that we are in a place where sequencing of the relapse can guide therapy. And as I come back to, we still have a long way to go to prove that we can impact patient care, cure patients based on this. So, in my opinion, we are getting very close to the time where all children with neuroblastoma, all children with cancer, should be offered sequencing of the tumor and their germline DNA at diagnosis and at any point in therapy where the tumor can be accessed. And there's a lot of hype about so-called liquid biopsies, and due to time, I'm not going to get into that today. We're happy to be addressing that. This is something that is an intense research effort in our laboratory. Is not ready for prime time by any close estimate, but it is something that will promise in the future be able to do a blood sampling and get all of this DNA information, because tumor cells do break free and get into the bloodstream and detect it, new technology. So the other major set of obstacles, pitfalls, is that it's not as simple as whether or not your gene has a mutation or not. We in pediatrics, because thankfully childhood cancer is rare, try to draw a lot of information from the adult community. But it's very clear, for example, that a mutation in this gene, BRAF, responds to BRAF-directed drugs very differently if it's a melanoma patient versus a colon cancer patient. So if a drug works against a specific mutation in adult cancer, does not mean that it's going to work in neuroblastoma. And that's something we have to be very cognizant of. Even more importantly, and I'll show you on, I think, the next slide, is that the different mutations, it's not just one mutation in the gene, that there are a whole series of mutations, and they they can be very different in their ability to drive a cancer or to be able to be targeted. And I'll show you that on the next slide. And then finally, and I'll come back to this, in the pediatric cancer community, in the cancer research community as a whole, we really have a deficit in people who are trying to study whether or not these DNA changes, whether these nominated targets can actually be effectively treated in the right preclinical models, in the right laboratory experiments before they come to children. I am of the strong opinion, as I'll get into, that children should not be the initial testing ground. There is urgency to get these drugs to children, but we need evidence that these work, and that's something that we have to work very hard to have a better pipeline to test this opportunities. So this is what I meant by ALK. So this is a picture of the protein anti-plastic kinase, and each of these dots are different mutations that have been discovered by Dr. Mose and her group. So there are a lot of different ways to take this protein that is normally in the off position and turn it on. So each of these different mutations turn it on. And what her laboratory has shown, this bar diagram here, is just the higher the bar, the more turned on it is. And so the red mutations make this protein really make the cells divide and keep on growing and growing and growing, more so than the blue and the green mutations. And why this is important is that the drugs that we have can effectively knock things down when they're about 100 or below. But these higher mutations are not sensitive to current drugs such as crizotinib. But thankfully, due to intensive research, there are newer drugs that can knock down these more hyperactivated mutations. So the point of this for you in the audience is that it's unfortunately not as simple as ALK mutation, yes or no. You understand the exact mutation in this gene or other genes and how they could potentially be targeted. So I've mentioned a few times the term preclinical data. And what I mean by this is the laboratory experiments that provide the proof of concept for clinical trial. While we are trying to personalize medicine, we still need to do this in the context of clinical research if we're ever going to be able to help children in the future and really be able to understand this. So preclinical data is generally a series of in vitro, which is test tube, or in vivo, which is generally mouse, although there are other animal models. And these are designed to try to recapitulate what's happening in the clinical setting. So in my laboratory and other investigators in the room, we have areas of our research laboratory that are set up like hospitals for mice, and we run clinical trials in mice. And it's my opinion, and this can be debated, that these data are essential to make the right decisions of what should be tested in children or what has the potential to help children in real time. And so this is, I'm just going to show two examples, and this is, again, a complicated slide that I'll try to make simple. So listed here are a bunch of drugs along the, target and then drugs along the y-axis, or the going up and down, and the same thing along the bottom. And the bigger the circle means that the combination of this drug and this drug is very effective in a test tube. And these are the sorts of screening that our laboratory is very engaged in, and I think others, because we know that single agents do not cure cancer. We've known that since the 1970s. So a big part of our program is that, yes, we have a hit, that ALK perhaps is a good target, but what is the right combination? So I think these kind of screens and looking for the big circles and drugs that be potentially combined and not be too toxic and combined is a big part of how we think that we need to make a difference in children soon, that children in particular, these new drugs should be brought forward in combinations, not as single agents. And then when you do this sort of work, and again, this is a complicated data slide, but I hope to make it simple. This is just tumors growing in mice, and this is without treatment, the tumors grow very quickly. And this is one drug, doesn't do much by itself. This is the other drug, doesn't do much. And the combination that came from this experiment, so picking the combination based on this, you can make these tumors go away and cure the mice. And so these are just different cell lines. This one, which has been renamed Felix for a UK patient whose parents wanted this cell line, who came from the patient, named after their child. If we would have treated this child with any of these single agents, this would have just not worked or worked only temporarily, whereas the combination is very, very effective and gives us promise that these combinations will be effective in children with the disease. So the take-home message from this is I want all of you to be very wary of clinical trials where the investigator cannot give you an explanation of what went into the design of the trial, why the drugs were picked, and what is the evidence that they could work. And I want you all to be very wary of clinical trials that sound too good to be true, because usually that's the case. And so some trials are based on so-called taking the tumor, putting it in culture very briefly, and in these so-called mouse systems that are just not proven to give us useful information yet. So like anything in life, if it sounds too good to be true, it probably is. And then I just wanted to end by talking about a clinical trial that will be opening at the Children's Hospital of Philadelphia that we hope to export to other hospitals soon, which we think is a relative first in the field. It's got a kind of a funky name, Nepenthe, which we chose because it is an ancient Greek word for a drug to take away pain and suffering, but it's a next generation personalized neuroblastoma therapy. And it will be the first clinical trial that we're aware of for neuroblastoma where it will start with a biopsy. So children with relapsed or refractory neuroblastoma will get a biopsy, their tumor, and sequencing. We have a system that can turn around these results in 10 days. Then they'll be assigned to one of three groups. And these are designed to be combination therapies. These two drugs for group 1, these two drugs for group 2, and I'll show you how we decide that in a second. And then for patients that don't match to this because they don't have the right mutations, there will be a standard phase 1 therapy that I'll show you on the next slide. We think only about 5 or 10% of patients who go through this algorithm will not be assigned to any of these treatments based upon the work led up to this trial. And this is the way it will work. It's a relatively simple flow diagram that will get the result. If the patient's tumor has an ALK mutation, we'll go into group 1, which is a combination that I showed you on those slides of the mouse growth curves of an ALK inhibitor and a so-called CDK6, CDK4 inhibitor. If this is not there, and if there's a mutation in a set of genes, MAP kinase pathway, they go to group 2. If those aren't there, and you have a mutation in something called the CDK genes, and something called the retinoblastoma gene is not mutated, you go to group 2. And then in none of those, you go to group 3. And so I don't expect you to know what this alphabet soup means, but the important point here is that many of these personalized cancer therapies or clinical trials not have a protocol-based assignment. It's left up to physician choice, which we think is too arbitrary. We think that this mechanism will allow us to, number one, help patients in the short term, but also learn about what we're doing right and what we're not doing right. So I'm just going to end with a couple of things. You had a whole section this morning on immunotherapy, and immunotherapy is personalized medicine. And I think that future immunotherapy trials will be based on sequencing results or protein results, in that patients will be assigned to immunotherapy trials, just like I showed for small molecule DNA trials, based upon whether or not the target is there. GD2 is usually there, but not always, and we need to be more precise in understanding what is there that we're targeting with immunotherapy. And again, I won't get into this because of time, but we have a lot that we can do in terms of predicting who's at risk for hearing loss, who's at risk for neurotoxicity, how we might modify standard of care drugs. And I think that you know, what we really see is that there's a lot of promise, but it's an urgent need to make sure that this is all worth it, that this really can deliver on the promise, and we think we need clinical trials like the Nepenthe. We need more of those where we can really understand whether or not biopsy and sequencing really results in cures. It's not in a vacuum. It's part of the overall package that we can bring to patients, but I think it's an important part of our changing paradigm. And as I prophesize about, we really need to focus our philanthropic efforts on really supporting the right translational research programs around the world and getting them to collaborate and to be able to move forward with some alacrity and some urgency.